Okay everyone, so this is the first part of Unit 2 of High Biology, Metabolism and Survival, and we begin by looking at metabolism and enzymes. Now, cell metabolism is just a term used to describe all the different chemical reactions that are involved in keeping cells alive, and of course many of these are catalyzed by enzymes. There are basically two different types of metabolic pathway. Catabolic pathways, first of all, take large complex molecules and break them down into smaller molecules, and energy is released. Anabolic pathways, however, take small molecules and combine them together to make large complex molecules, but this requires energy. Catabolic and anabolic pathways are often linked. For example, respiration, the breakdown of glucose with oxygen, releases energy which can be used to synthesize proteins from amino acids. Poisons, toxins and venoms, they can often disrupt metabolic pathways. Metabolic pathways, they often have reversible and irreversible steps, and a good example of this is glycolysis, the breakdown of glucose to pyruvate in the cytoplasm of cells, this being the first part of aerobic respiration. As an example, if we have a look at glucose, it's broken down by enzyme A into an intermediate substance, intermediate 1. Enzyme B, however, this is an irreversible enzyme that can interchange between intermediate 1 and intermediate 2. If too much intermediate 2 is made, it can be converted back into intermediate 1, so it can follow alternative routes. That being, intermediate 1 can be used to create starch in plants, or glycogen if it's in animals. However, the production of intermediate 3 by enzyme C, that's referred to as a key regulatory point. From that point, the reaction must proceed on to the production of pyruvate at the end of glycolysis. However, occasionally, in the case of glycolysis, if there's a plentiful supply of glucose to begin with, then often alternative routes can be carried out instead. Bypassing the reactions of enzymes A, B and C, glucose is converted into an intermediate substance, sorbitol, which takes a more direct pathway to the production of pyruvate at the end of glycolysis. Now, many of these metabolic reactions take place on membranes, and the membranes, of course, separate cell contents from either the extracellular environment or within the cell, compartmentalizing different reactions for different purposes. Basic cell membrane structure is, of course, a phospholipid bilayer, which you should be already familiar with. Each phospholipid having a hydrophilic head and a couple of hydrophobic tails. But within the membranes, of course, there are various proteins, and each of these have certain functions, such as they can provide structural support, they can create channels allowing the transport of small molecules, they can form carriers, which are involved in active transport, pumping substances across the membrane against the concentration gradient. Some of them can be enzymes, of course, catalyzing various reactions, receptors for hormones, for example, and as antigenic markers for cell identification. Now, if you have just a brief look at different types of molecular transport, these should all be really familiar to you already. Passive transport, such as diffusion osmosis, normal molecular movement, they do not require energy. And of course, diffusion is the movement of molecules from an area of high concentration to low concentration down a concentration gradient, as it were, like a ball going down a hill. Active transport involves the movement of particles against the concentration gradient, of course, and it requires energy in the form of ATP, which we'll look at in more detail later on. And a good example is the sodium potassium pump, which is particularly important in muscle and nerve cells. Another membrane enzyme that you're going to be very familiar with later on is one called ATP synthase, which is involved in the production of ATP in the final stages of respiration, like so, and it involves the movement of hydrogen ions past or through the enzyme, which catalyzes the production of ATP from ADP and inorganic phosphate. Okay, so let's have a look at enzyme action in terms of activation energy and chemical reactions in general. Because for a chemical reaction to occur, the reactants often need activation energy, at which point, if they receive this energy, they enter a transition state, and then the reaction will proceed to create the products. Of course, enzymes lower that activation energy, and in this way helps to speed up the chemical reaction. And of course, as you know already, enzymes take part in the chemical reactions, but they remain unchanged, so you can use them again and again. Now, the reason we have enzymes in our bodies, and organisms have enzymes in general, is because normally for a chemical reaction to proceed at a rate fast enough to maintain life, it would normally, under other circumstances, be required to occur at a very high temperature. Of course, that would destroy cells, so that's why we have enzymes. So the reactions can proceed fast enough to keep us alive, but without destroying the cells, so at a lower temperature. Some basic background knowledge of enzymes now that you're already familiar with, I'm sure. That, of course, enzymes are proteins. They are specific to certain substances. Each specific enzyme has an active site, which is complementary to a specific substance or substrate. But in more detail, what actually happens when the substrate comes into contact with the enzyme, the active site undergoes what's called an induced fit. So it kind of wraps itself around the substrate. So when the substrate comes along, the active site forms a very tight, snug fit with that substrate. The active site also is involved in the correct orientation of substrates. As an example, we have a couple of substrates coming in. Induced fit occurs at the active site, and of course the active site, once the substrates are in contact with each other and the active site, the reaction is catalyzed and the product is made. The product has a lower affinity to the active site than these substrates do, and so it's released, so the enzyme can go on and do exactly the same again. Factors affecting enzyme action, of course, we've all come across these. A suitable temperature, of course, an appropriate pH, and an adequate supply of the substrate. But what we'll look at later is that inhibitors will slow down enzyme control reactions or bring them to a halt altogether. In terms of substrate concentration, with increasing substrate concentration, there's an increasing rate of reaction. However, the reaction rate levels off, and that's when all the active sites of all the enzymes present are being used simultaneously. Okay? So any further increase in substrate concentration won't increase the rate of reaction. And so at that point, enzyme concentration has become a limiting factor for that reaction. Enzymes, of course, as we have seen already, can carry out reversible and irreversible reactions, and the direction of a reaction will depend on the concentrations of reactants and products. Okay, thank you very much indeed.